Happy New Year! Unbelievable. Here we go. Let's start with Mendelian genetics. We're going to start here with the man with the plan. That would be Gregor Mendel. Back in 1850, this man did some work. He's considered to be the father of genetics. That's an important thing to say because that is absolutely a standardized question that's asked quite frequently. So the father of genetics is Gregor Mendel. And old Gregor, he was a monk that worked with pea plants. And back in 1850, there were two ways to do science. You either were independently wealthy, and so you used your own money. You didn't have to work, and so you could conduct research, or you went into the monastery, believe it or not. And so that's where Gregor was. And as relationship to character and trait, character is an inheritable physical feature. So this is a characteristic such as eye color or hair color. So it's inheritable. All right. A trait, oh, well, let me go back here. A trait requires inheriting two alleles, one from each parent. So, and this is a variation of the character, such as blue eyes or black colored hair, something along those lines. So the character is the actual physical feature that's inherited, and the trait is requiring two alleles, one from each parent, one from mom, one from dad. All right. Now, when we talk about this word alleles right here, alleles are different versions of a gene, just like it says here. So remember, a gene is a distinct DNA sequence that's going to make one protein or one enzyme. So we're talking about brown, blue, green eye color. There's three different versions or DNA sequences of a single gene, but they're all making eye color. So there's going to be a different sequence that's going to code for that particular color, but it's going to be, it's still for eye color. So each trait, when we talk about alleles, each trait needs two alleles, one from each parent, so that that gene can be expressed. Dominant alleles are given, are given a capital letter. Oh, it's not here. Okay, so this is not really a great example with a P, but this is a capital letter P. That's one allele. Another allele is a lowercase p. If it's a capital, it's dominant. So we write dominant alleles in capital letters. And these are like books or recipe cards with information in them. So if the dominant allele is present, that's what you're going to see. That trait is going to be expressed. And it would be kind of like the recipe card from the whole book, or one recipe from the recipe book. The recessive alleles are given lowercase letters. So these are like books or recipe cards with blank pages. So there's no information on them on how to make the protein or enzyme. That actual blueprint is missing. All right. And so lowercase letter, recessive, uppercase letter, dominant. If the dominant is present, you're going to see that trait. If the only way you're going to see the trait, whatever the trait is of the recessive gene, the only way you're going to see that is if there's no dominance. Like here you can see there's a capital letter, so these are purple flowers, but there's no capital letter here, there's no dominance, so then we're able to see that recessive trait. All right, let's see. All right, so we get one from each parent, and that means one is in the sperm and one is in the egg. All right, let's look at the concept here of true breed versus hybrid. Now this is a pretty big deal for me, especially with the birth of my puppies. They're true breeds. So when people ask me, when they talk to me about buying a puppy, they talk to me about, is it a purebred? Is it papered? Does it have a certificate? And what they're asking is it a true breed? Is it a lab and a lab, a pure lab and a pure lab? Or if I took a lab and I mixed it with a German Shepherd, then we're going to have a hybrid. All right, so true breed versus hybrid. And I want to pause here for a quick joke. Why did the chicken cross the road? Or no, I'm sorry, I totally blew that. What, what did the chicken say when it crossed the road? It didn't say anything. Chickens don't talk, goobers. All right, so true breed. So a true breed, these organisms have only one type of allele for that trait. So like I said, if we're talking about a Labrador, it's only mixing with a lab. But when we're looking at what's called the genotype, that would be big B, big B. So if we were looking at a true breed, then this would be 2 capital letters or 2 lowercase letters. So you could have a big B, or a little b, or a big R, and a little r. Those are true breeds. They're also known as, this is an important word, homozygous alleles. Now remember from our prefixes, homo means what? It means same. So what this is saying is both of those alleles are the same. They're either both capitals or they're lowercase. All right, so those are homozygous. With a hybrid, let's not go there yet. With a hybrid, you're creating an organism with 2 different type of alleles. So that would be like a big B, little b. So hetero means different. And so when we're talking about if it's heterozygous, that means it's going to have a capital letter and a lowercase. It's got 1 dominant allele and 1 recessive allele. All right, so now we can move on to this next one. We're going to look at right here where we take a true breeding purple and white and why we end up with this. We're going to look at the Punnett square coming up. All right, phenotype versus genotype. The way I like to remember this is that phenotype sounds like it's beginning with an F. Phenotype, and I like to say phenotype face. The phenotype is what you're actually seeing. It's referring to the physical trait that you can see. If I say someone has blue eyes, or they have blonde hair, or they have type O blood, we're referring to the phenotype at that point, okay? So phenotype, face. That's the easy way to remember it. Versus the genotype is the genetic. It's the genes. This refers to the organism's genetic makeup. So when we say big B, or big B, or little b, we're referring to the genotype. So if we're talking about the letters, we're talking about the genotype. If we say what trait that codes for, then we're talking about the phenotype. So red hair, phenotype. Okay. If a genotype of an organism is unknown, then we perform what's called a test cross. Now this is fascinating. I'm going to show you this, or I might have already shown you this in class of my puppies and how we ended up with the chocolate labs. But we do a test cross, and in order to do a test cross, we use a homozygous recessive. So like a little b, we're going to use a homozygous recessive to mate with our unknown. So what that does is it allows for no information to be covered up by some known dominant allele. We're going to do this cross in class so that you can see. But that's how you do a test cross to find out whether you have pure breeds or whether you have hybrids. Oh, here we go. Oh, goodness. Oh, boy, here we go. All right, so we want to find out, is this parent homozygous dominant or heterozygous? And this one is obviously homozygous recessive. So we use this as a test cross, so we know what this is. When we cross them, if we have big P, then all our results are going to be purple because the purple's going to mask the white. But if this parent is big P, little p, if it's heterozygous, then we are going to end up with some parents here, some of the offspring that can be white. All right, if there's any white at all, then that tells us the big P that we have heterozygous. That's how when we crossed my black lab and the yellow lab, we ended up with chocolate labs. And I'll go into the detail on that coming up. But all right. Now, Punnett squares. The Punnett square, what the Punnett square does, oh, people love doing Punnett squares, but what the Punnett square does is it shows the possible genotypic outcomes. What are the possible genotypic outcomes? That doesn't mean that if we get 4 offspring here, that's what each one's going to look differently. It's not. It just tells us what are the possibilities. So this chart here displays 1 in what's called a monohybrid. That makes sense. Mono means 1. So we're crossing 1. We're just looking at 1 trait. Dihybrid, we're looking at 2 traits. See, there's 2 letters here. And so our results are, we're going to have 16 possible outcomes. So if we go back here, 4 to the 1, monohybrid, 1. 4 to the 1 is 4 to the 1 power. 4 to the 2 power, because we have dihybrid, there's 2, so 4 to the 2nd power is 16 squares, 4 times 4. If we go to, oh, we don't have the 3rd one up here as an example, but if you had 3, it'd be 4 to the 3rd power, because there's 3 different traits. How many would we have? How many possible? So 4 to the 3rd is going to give us how many squares? Okay, figure that one out, put that in your notes. All right. Now, Mendel's law of segregation. To segregate means to separate. That means that alleles on homologous chromosomes, so chromosomes that are the same, so they are connected together. So alleles on these homologous chromosomes move independently. Law of segregation, they're going to move independently of one another. They walk to the opposite poles of the germ cell during anaphase 1. So this separation is continued through anaphase 2, and so they're going to be pulled apart. Okay? The law of independent assortment, let's see. The law of independent assortment describes the behavior of pairs of homologous chromosomes during metaphase 1. This means that they're going to sort independently. They can go in different directions, not based on, so here's independent assortment. They can sort in any direction. They are not tied to each other. Oh, I just have to tell you, I am so in love with my puppies. Sorry, a little distracted, but I am just so in love with these puppies. Okay, so back to the independent assortment. Each pair of homologous chromosomes moves to the metaphase plate during metaphase 1. And remember our stages, PMAT, prophase, metaphase, anaphase, telophase. I think we're going to stop for today on that, and we'll pick it up probability in the next one.